Actually, it's not all about what you do when you have local melanoma or what happens when you're just initially diagnosed, because our real issues now are what happens when standard initial therapies don't work optimally for you. And that's where a lot of the work that is now standard has come from. It's clinical trials, it's understanding the options, it's understanding our targeted therapies, etc. And there's no one better at that than Dr. Sullivan. So, Dr. Sullivan, please.
Thank you very much, Dr. Hamid. It's nice to have somebody finally say something nice about me. Right. So my task is to do exactly what Dr. Hamid said. By the way, thank you to Dr. Hamid and to the Angeles Clinic for the invitation. Thanks to Connie for all the work that she's done to arrange a smooth arrival and departure at some point. I'm going to talk about clinical trials, and I'm not going to talk about every clinical trial that's out there because we'd be here all weekend and maybe all week, but very select clinical trials. Here are my relevant disclosures. One thing I was thinking about during Dr. Patel's talk is that a synonym for disgusting would be gross, not grossing. But she mentioned the word grossing, for doing stuff with samples. But why is that? It's not really a disclosure. It's really just a comment.
English is my second language.
I mean, I don't think English makes sense to anybody. Okay, so Dr. Hamid showed a version of this earlier, but this is where we are in 2024. And so when we begin to think about what are we going to do clinical trials, and it's important to see where we've come from to help understand where we're going. And so I think the summary is a lot has been done and accomplished over the last 15 years, but yet we still have a ways to go. And so when we begin, again, to think about what should we be studying, what patients should we be running and enrolling onto clinical trials, well, first of all, it's important to know that almost all that data, not all that data, but almost all that data to lead to all those approvals are in patients who had never been treated with the therapy for their advanced melanoma before, and almost all of them had cutaneous melanoma. And so, again, it's important to think, okay, well, where could we get better? Well, we could have better frontline therapies, and Dr. Hamid did a very nice job of describing what our frontline standard of care is and also some of the studies that are being done there. We need better second and third line therapies after CTLA-4, after PD-1, after LAG-3 inhibition. And we need better therapy for uveal, acral, and mucosal melanoma, which actually Dr. Hamid did a great job of taking us through. I'm going to tell one little story about frontline therapy. It actually is a little bit of a reinforcement of one of the things that Dr. Hamid was saying, but I think it's that important to talk about. So I'm a big Talking Heads fan. I don't know if you guys have heard of them. They're not really an LA band. You know, they're from the East Coast. They went to RISD, which is a Rhode Island School of Design, and they were hanging out in New York City for the 70s. They may or may not have become famous in the 80s. But one of their big songs was Burning Down the House. And the first line is, I'm not going to sing it for you. I'll just, but watch out. You might get what you're after. And when we think about immune checkpoint inhibitors, we are trying to harness the immune system for good. And I think it's a lot like fire. Like fire is what separates us from the other great apes, right? The harnessing of fire changed society, or created the opportunity for society. But if it goes awry, bad things happen. And so when we think about how can immune therapy go wrong, I tend to think of it in the setting of this slide. And so immune checkpoint inhibitors can be dangerous. Other dangerous things are patients who give you things like this. So this is a Consumer Reports, and my patient saw this, and it says, what you know about what you don't know about your doctor could hurt you. And there's a picture of a doctor with a martini glass. I don't drink martinis. But in any event, he decided to cut out a picture of my face from the internet and paste it on that. I actually have this in my office because it's amazing. But I like to use it just to say where all the problems of immune checkpoint inhibitors can happen. And so, like lots of things can happen when we give immune checkpoint inhibitors. And what I counsel my patients that any tissue in the body can get inflamed. These are really remarkable drugs that are targeting something very, very basic in our body's regulation of our immune system. IRAEs is the short version of immune-related adverse events, which is the term we use to talk about toxicity. And they can be driven by numerous potential mechanisms, but almost all of them are related to loss of tolerance. And tolerance is important because our immune system can get activated. It gets activated to go attack stuff. Usually that's cells that are infected by viruses or tumors. Tolerance keeps us from being perpetually attacked by our own immune systems. And so if you lose tolerance, you're at risk for being perpetually attacked by your immune system. And so I think the central challenge of cancer immunotherapy is to try and sort out how we can separate the things that drive the immune system to attack cancer and the things that drive the immune system in the setting of these drugs to attack normal tissue. And so the question is this: can we uncouple anti-tumor immunity from anti-patient immunity? So you saw this before. This is data from these seminal clinical trials of either nivolumab, ipilimumab, or ipilimumab, nivolumab in patients with advanced melanoma haven't been treated before. And the summary of this is that this protein, this substance called interleukin-6, can drive bad outcomes. So if you have high levels of interleukin-6, you're less likely to have a response. If you have high levels of interleukin-6, you're more likely to have your tumor grow and you're more likely to die. And so IL-6, it turns out, is something we can target. And so one question in our minds is, can we target IL-6? The drug you heard of before, tocilizumab and sarilumab, actually block IL-6 receptor, so it's blocking the way that IL-6 signals. And so this is, one way that we can make frontline therapy better is reducing side effects. And you saw this before too. But this is, I think, going to end up being a really important clinical trial. The sort of proof of principle that you can give drugs not just to make response rates better, but to make toxicity better. The original data from this study was presented 3 years ago. This is going to be updated this year by Dr. Weber at ASCO in 3 weeks. And basically, the early data, which may or may not mirror the final data, is going to suggest that there indeed is a better response rate and a lower toxicity rate than we would have expected by these two drugs, Ipi and Nivo, given at these doses. The follow-up trial, which you also heard about, is looking at 3 drugs plus a fourth, and the fourth being the IL-6 receptor blocker, sarilumab. This started about a year ago. The first part of the study is just about done. Now the randomized part comes, and the randomized part's important because ultimately, if we want to take this idea to a larger stage with hundreds of patients to really see, can we redefine the standard of care for patients by targeting toxicity, you'd probably have to have some randomized data that says, no, we did a small randomized phase 2 trial. This approach looks really good. And so the primary endpoints, again, reduce toxicity, improve responses. So that's all I'm going to talk about frontline therapy. I know Dr. Hamid talked about it already, but I think it's really important that not only are we thinking about folding in drugs that might make it better to have more responses, but also we might be able to improve the risk-benefit ratio, which then ultimately can make these therapies better. All right, what else are the unmet needs? Well, again, I mentioned the second and third line therapy after standard of care immune checkpoint inhibitor therapy. We have a few different ways we can target this. We have targeted therapies targeting BRAF mutations and NRAS mutations, and we have new immune therapies, and we can cross over after those. I'm taking 2024 off from talking about BRAF targeted therapy. Spent most of 2023 talking about it. I'm just not doing it. But it doesn't mean I can't talk about NRAS targeted therapy, which ultimately it's like the same studies. So let's talk about NRAS. So BRAF and NRAS are proteins in this pathway called the MAPK pathway. MAPK is actually otherwise known as ERK, which is sort of at the bottom of that pathway. And about half of patients with melanoma have a BRAF mutation, and about a quarter of patients have an NRAS mutation, and it activates this pathway, and we actually have drugs that can block this pathway. And so this is one potential therapeutic option for patients. And basically, the strategy is just hit these proteins with big hammers, or really little hammers, so we call them small molecules, but basically big hammers. So there's emerging data with BRAF dimer, MEK, and combinations of these in NRAS mutant melanoma. So BRAF dimer, so on this slide, ignore the man with the large hammer for just a moment, but you see the red RAF, and it's kind of signaling, there's three arrows going to this MEK protein. That's the BRAF that's typically mutated in melanoma. Amazingly, it just signals all by itself. Usually what RAF does is it binds with another RAF through the protein called RAS. So RAS binds to two RAFs and signals. And so a RAF dimer is something that blocks those two RAFs stuck together, as opposed to that red RAF. I don't know what color they are in people, but on the slide, bright red and brown. And so RAF dimers and MEK inhibitors have been tried in NRAS mutant melanoma. So on the left, this is one MEK inhibitor called binimetinib in data from 2 trials. That's a waterfall plot, so down is good, and you can see that most patients have down as their best response. And so that was in 2013, and people were like, wow, we might have a targeted therapy. You'd already had vemurafenib, and dabrafenib was coming out, and we're like, we're going to have binimetinib for NRAS mutant patients. Let's do a trial, and a trial was done, and that's the lower left, which is binimetinib versus dacarbazine. And it was technically a positive trial, but not positive enough for the FDA and other regulatory agencies to say, yeah, we should approve this for patients. And so it never got approved, but it clearly has some benefit in this patient population. It's just not enough. This is some work with a couple of different RAF dimers. Now, these are all solid tumor studies, but there are patients with melanoma on both the RAF dimer, the above one and the below one. And most patients who are going on this, their tumors are getting a little bit smaller. And when you start combining it with the 2 types, we're beginning to see additional responses. And so based on all of this preliminary work with these types of drugs, this trial was launched. So this is a trial of a RAF dimer inhibitor, which is designated by the blue star with the yellow stuff around it. And that's called naporafenib. So any RAF inhibitor will always have the word RAF in the middle of it. And then it's either combining with a MEK inhibitor called trametinib or an ERK inhibitor called rinaturkib. Why trametinib isn't tramekinib, I don't know. But again, words are problematic for me. Still trying to wrap my head around gross, grossing, and disgusting. So the problem when you block this pathway in two ways is you get a lot of side effects, and almost everybody got a bad rash. And it turns out you can give medications to try and prevent the rash either from happening or from being as bad. And so that's one thing that's happened as part of the study. But if you look at the bottom right and sort of furthest right are the NRAS mutant patients, there's a lot of down in those waterfall plots. And individually, there's more down than we usually see, meaning the responses, the regressions are more profound than we've seen with other things. And so based off of this data, there's now a randomized trial that's been launched that's looking at two doses. So the pink and the light blue doses, because those are two different doses of naporafenib and trametinib. So they're being studied against each other in the first part of the study. The third arm of that against each other is trametinib by itself. So we know that MEK inhibitors by themselves have some benefit. Is that me? No, good. And so that's cool. And then once that goes forward, we're going to pick a dose. So whatever's the best dose of the combo will move forward, and that'll be decided based on responses, be decided based on toxicity, and then there'll be a physician's choice arm. And if you're really interested, you can go on chemotherapy, or you could also go on trametinib. I know what I'm going to choose. It won't be chemo. But in any event, I like this trial. We're excited to get this up and running. And it'll be the first trial of its kind testing two drugs in this randomized way, because if this trial works, this would be a new therapy for patients with NRAS mutant melanoma. So targeting the MAP kinase pathway is the way to go for now. Emerging data with dual pathway blockade, RAS directed. So actually, none of this is targeting RAS itself. RAS inhibitors are being developed now. And so this hasn't really been targeting NRAS to date, but we're hopeful that those types of therapies will also be developed over the next several years. And for now, the best we're going to do is this randomized trial, which we're excited about. Okay, coming down the home stretch, I have 3 minutes. I got this. No, I don't need any more. All right, so this is, now we're going to talk about injecting tumors. There is one injectable therapy approved called TVEC, or herpes light, as Dr. Patel called it earlier today. Going to use that forever. So this is a drug called mRNA 2752. It's from Moderna. It's an mRNA. It's actually 3 mRNAs wrapped up in a lipid nanoparticle that are, when it gets injected into the tumor, these proteins get expressed. And the proteins are called OX40 ligand, IL-23, and IL-36 gamma. And the idea is to improve the interaction of the immune system so that it can be more capable of attacking the tumor. There were a couple parts of this trial. Part A was looking at just the drug by itself. Part B was giving it with a PD-L1 inhibitor. On the left is one of my patients. Amazingly, in the upper left of the left, that's pretreatment. It doesn't look that bad, but he had like lots of bumps. And we started injecting these bumps, and they became dark. Because remember that picture of the melanophages from Dr. Patel's talk? So when you inject a tumor and the tumor cells explode, all these cells come by and they like basically eat up the pigment. That's actually how tattoos work. And so you can actually see tattooing of the skin from melanomas that are being destroyed. So that's what this patient had. And so on the lower left is viable tumor. On the lower right is death of tumor. And then all told, this hasn't been remarkably effective, although there is a melanoma-specific cohort that'll be presented sometime in the next year, which I think is going to look better than this slide looks. And on the lower right, it's basically just saying when you inject tumor and you look before you inject and after you inject, you see more activity of the immune system in the patients whose tumors got smaller or didn't grow versus the ones that didn't get smaller and grew. Okay, any other cool ideas? Yeah, cryoablation is cool, literally and figuratively. You stick a probe into a tumor, you cool or freeze that tumor, and you thaw, then you freeze, and you thaw, then you freeze. I don't know how many times you freeze thaw, but several times. And it leads to interesting immune activity. We ran a trial led by Megan Meridian, one of our medical oncologists, and Florian Fintelman, one of our interventional radiologists. And we treated 17 patients who were on immune therapy. Their tumors were growing. We kept them on that immune therapy, and we just cooled or froze one of their tumors. And then when we measured benefit, we didn't measure the tumor we froze. We presumed that that was going to get smaller. So when you see 23.5% response rate, that means that those patients' tumors outside of what were frozen got smaller. So almost a quarter of patients' tumors got smaller, enough to call that patient a responder. And about half of patients had at least 6 months of durable disease control beyond when they got their tumor frozen. And again, these were patients who were progressing on the same drug we continued. So I think pretty compelling. Thankfully, Nature Communications thought it was pretty compelling too, because it just got published. So we're really excited about this. I may skip this for time. But basically, in terms of tumor-directed therapy, lots of things you can inject into a tumor. mRNA 2752 looks interesting. Certainly some very cool things happening in tumors. Hopefully the melanoma-specific cohort looks good. With cryo, responses in about a quarter, disease control in about half. And we now have a follow-up trial in patients whose tumors have progressed on initial immunotherapy. As long as they didn't get ipilimumab before, we're giving ipilimumab, nivolumab, and we're cryoing, and we're going to see what happens. And so we've enrolled about 8 to 10 patients on that study. I think we're aiming to enroll about 30 or 40. With that, I will thank you. And you've seen some of these pictures. I'm not sure why everything was so red and pink in the lower right picture, but I refuse to have my picture taken with Dr. Patel any further, because she's probably going to show it in another talk. With that, I thank you and look forward to the questions later.